All right, thank you very much. Who's ready for Moose Part 2? Once again, my name is Elias Rosenblatt. I'm a PhD student with Vermont Cooperative Fish and Wildlife Research Unit. And I'd like to acknowledge my committee members, especially my advisors, Dr. Terry Donovan and Dr. Jed Murdoch, as well as Stephanie McKay and Dr. Katie Geeter from Vermont Fish and Wildlife. And today I'd like to talk about some preliminary findings we have about moose population genetics here in Vermont, so some fun things. So moose are really emblematic of the northern boreal forest, and they're incredibly important for ecological, economical, and societal systems all across North America. However, many populations, especially here in eastern North America, are facing some problems Josh just highlighted. At the same time, we know that the maintenance of genetic diversity is incredibly important for population persistence. However, we don't really know much about moose population genetics at all in this area of the world. So, before I dive into what we've learned about moose population genetics here, we really need to look back into ancient history, and actually not that long ago. Moose arrived here in North America only within the past 15,000 years. They crossed the Bering Land Bridge, and they were stuck in southwestern Alaska for a couple thousand years until the glaciers from the last ice age began to recede, and moose were able to work their way along, radiating eastward across North America, arriving on the eastern seaboard of North America at the very end. And an important consideration here is that when moose, when this radiation happened, this wasn't some grand migration of tens of thousands of moose setting forth following the great glaciers. It was more of a trickle of dispersing individuals. And so these populations were set up with this radiation by only a few individuals. That's what we think. We weren't there. However, from a genetic standpoint, this kind of raises some red flags here. There were likely founder effects where these small groups of animals that started populations resulted in these populations having very low genetic diversity. Coupling with that, there were probably some hard times for these populations. They probably went through some population bottlenecks where there was a reduction in the number of individuals in a population, which also reduces the genetic diversity in those populations. Unfortunately, recent history, as Josh just presented about, or mentioned in his previous presentation, recent history hasn't been too kind to the moose, mainly through the over and unregulated harvest of moose and other wildlife, a booming timber industry, and mass land conversion from forests to areas for livestock and agriculture. We really lost our moose at the turn of the 20th century. And those moose were gone for a while. They came back and we're doing really, really well. And as Josh touched on, we now have this problem. We have this tick called the winter tick, a deer family-specific tick that is causing elevated juvenile mortality rates, which are causing population declines across much of eastern North America. And so with all of this history in mind, we can now start thinking about the genetics. And last year, there was a paper published by Kalfleisch and others, including two of my committee members, that in part looked at the genetics of 4 moose across their distribution in North America, ranging from Alaska to Lowell, Vermont. And they were doing a bunch of other things in this paper, but they compared a measure of genetic diversity across these 4 animals from Alaska, Wyoming, Idaho, and Vermont. And this y-axis, basically, the higher you are, the more genetic diversity you are carrying as an individual. And so what they found was that Vermont had the lowest genetic diversity, the Vermont moose, I should say. Again, these are only 4 animals, very small sample size, but this sparks the question, what's going on in Vermont? Is this just an anomaly, or is this a pattern that we might want to know about? Because again, genetic diversity is important. So that kind of does set up the challenge here, and what I'm setting out to do tonight is to talk about how we can improve our understanding of moose population genetics in Vermont. And I'm going to walk through how we've been using extensive sampling of moose across Vermont to explore the genetic diversity of the herd here, the level of inbreeding, and the relatedness between individuals in the state. So for sample collection, we collected genetic material from 152 individuals across the state, and these individuals were sampled in 2 ways. First off, they were lucky enough to be involved in our radio collaring operation in the Northeast Kingdom. If you're not familiar, this is our study area in the Northeast Kingdom, that great polygon. These animals were radio-collared as part of a larger survival and reproductive study conducted by Vermont Fish and Wildlife, and all the capture, animal restraint, on all that was followed UVM Iacocca animal care protocol. So we got a nice sample from those individuals, and then the other batch of animals were those that were legally harvested through Vermont's legal moose hunt every fall. And so hunters would bring their moose to a hunter check station, and Vermont Fish and Wildlife staff, I believe that's Tony Smith right there, would grab tissue and hair from those animals, and we could extract DNA from those materials. And so our sample distribution for these harvested animals are these green blobs on this map of Vermont. These greenish blobs are towns where moose were harvested, and the darker the green, the more number of moose were harvested there. So you can see we have a distribution of animals across the state here. All right, so once we had the materials in hand, we, is a generous term, other people would clip off the nice tissue that had the genetic material, and then the DNA was extracted and amplified, and each animal was genotyped using a single nucleotide polymorphism parentage panel, or a SNP parentage panel. If you're not familiar with SNPs, they are variable base pairs in DNA that vary between individuals and allow us to track who's related to whom, where animals are from, etc. And so for each animal, we calculated the heterozygosity ratio, and that is a measure of genetic diversity, the identity by state, and that is the proportion of genetic material that is shared, or that is in common between individuals, the level of inbreeding, and identify related animals in our sample set. So for some results here, for genetic diversity, just to orient you, on this y-axis is our heterozygosity ratio. Again, the higher up you are, the more diversity you contain as an individual. And for reference, this is that moose from Lowell, Vermont, in the paper that I referenced earlier that sparked this idea that we may have low genetic diversity here. Well, these are all the other moose, all those dots. So actually, heterozygosity ratios in Vermont are low, and lower than we thought. These are lower than the individual that sparked this question. When we looked at identity by state measures, we got an average measure of 0.74, and in plain English, that means that 74% of genotypes are shared across sampled individuals, regardless of whether or not they are related or not. That's really high. In the human population, that number's around 10%, somewhere down there. So, animals have low diversity in general, and also they share a lot of the same information. Now, moving on to inbreeding. Our y-axis here is a measure of excess of homozygotes. Basically, if you as an individual moose, one of these black dots, are below this blue line, that means you have some level of inbreeding going on in your system. And so over half of our study animals have an excess of homozygosity and show signs of inbreeding. Finally, moving on to relatedness. Here are our results. I apologize for the really bad map. I don't really know how to display this information any better. We had 46 animals, about 30% of our sample, that were related to one another. And these animals were identified across both harvested animals and radio-collared animals, as well as across wide areas of Vermont in northern and central Vermont. These black lines indicate related pairs. And so the longest line here is 106 kilometers. So this conforms with our understanding of how moose disperse when they're young and establish home ranges. They do move large distances. So this is a neat application of these genetic data. So in summary, we have preliminary evidence of low genetic diversity. We have evidence of inbreeding in many sampled individuals, and we have related animals ranging across northern and central Vermont. Now, this is all very preliminary, and it's unclear whether these findings are driven by current inbreeding levels, recent history, all this stuff, or this is just a ghost of the establishment of the population. Remember, there were probably a series of founder effects and population bottlenecks that led to the establishment here in Vermont. So it's hard to say what is the cause of the patterns that we are seeing. However, these findings may have implications for the resilience of moose in Vermont in the face of changing forests and climate. The example that I want to go with here is that moose, and us as humans too, have genes that code for inflammatory and immune response. And thinking about winter tick as the primary thing that we're worried about, If we have low genetic diversity in those genes, that means that the array of genetic options for dealing with a parasite, we don't have a wide array of those tools. And by we, I mean the moose. In summary, and to wrap up, this is really the first real look of genetics in moose in Vermont. And here's a nice mom and calf hanging out. What this is going to set up is, for some more of my PhD research, is if this pattern is unique to Vermont, or is this a regional pattern? How does gene flow currently exist between Vermont and the rest of the region, as well as what are the implications for resiliency in the face of ticks, as dictated by genetics. So with that, I'd like to acknowledge a whole bunch of people, my graduate committee, all the collaborators who made all this genetics work possible, the Vermont Cooperative Fish and Wildlife Research Unit, Jake DeBow, Josh Bluen, for all being awesome team members, Cedric Alexander from Vermont Fish and Wildlife, who's the moose biologist, the rest of staff from Vermont Fish and Wildlife and cooperating moose hunters, Native Range Capture Services for doing the animal restraint and taking samples, and Silvio Conte National Wildlife Refuge staff for their support. I'd also like to acknowledge my support funding support from the Rubenstein School through the Stephen Rubenstein Graduate Fellowship, 2 USDA grants, including the NIFA McIntyre Stennis Project here, USGS Cooperative Research Units Program, as well as federal funding through Pittman Robinson for the radio collaring operation. So with that, I will take any questions.
Gene flow is not as, I guess, more like, it's like low, we have low gene flow because it's just a smaller space.
Yeah.
I think that like spatial has something to do with it, maybe?
Yeah, I think a combination of the mixing of populations around here, as well as just the recent resurgence of moose. Most of this area lost their moose until the 60s and 70s. And so where those moose came from, probably from northern Maine and Quebec, but as the connections reestablished, those helped fuel the current state of our population genetics, but I can't really say what lever is really resulting in what we're seeing here. But that spatial component absolutely is.
And it'd be really interesting to look at, I know it's like a COX-1 gene, like a mitochondrial gene.
Oh, yeah.
I know it's like super recent, but I don't know, I just thought about it.
Yeah. Great suggestion.
I think I have 4 questions, but I'll try and pick one. Do you think, I don't know if you have a sense of the management possibilities, but I'm just thinking about flying some new ones in somewhere else to expand the genetic. Do you think that's a possibility?
I think we need to do some more work before we advocate for that. It's certainly not a good sign, what the initial signals that we're seeing. But we are working to get some better, more detailed information into the moose genome as far as, okay, we see these broad measures, what are they really impacting? And I think after that, then you could advocate for something like that. But I would say translocating would make the most sense if you were to ever go down that route. Artificial insemination for moose would be very difficult, mainly because the breeding window is very, very small. And we still have the problem of winter ticks that would kill any resulting calf. Most likely, not absolutely, but that's a bottleneck they'd have to go through. Yeah, yes.
Well, so I think maybe you're going to get here, but with the combination of low genetic diversity and winter tick mortality, are you looking at whether more homozygous individuals might be more susceptible to winter tick?
Yes, absolutely. And so, yeah, I'll be looking at genetic diversity measures, a couple of them, and compare that. There are a couple hypothesized relationships between how genetically diverse you are as an individual and your ectoparasite load. So there are a couple previous works that I'm kind of bouncing off of.
I was just wondering, do the adults withstand the ticks better than the juveniles? Or
Yeah.
the picture we saw, the previous presentation looked like an adult?
No, that was actually a calf. Yeah, but it's a great question. The adults get hammered. They have 8, I think there was a bull in New Hampshire that had over 100,000 ticks or something like that. that certainly impacts them in a non-lethal way. It likely impacts their reproductive potential moving forward, but it doesn't kill them outright like it does with the calves.
Awesome, we're actually out of
Yeah.
time, but you can ask the last any question after. Thank you everybody for coming.
Thanks.